Okay, so welcome back to this next video in which we are discussing G protein coupled receptors. Okay, right, so in the previous video what we've just done is looked at the general features that all G protein coupled receptors share, and we've then talked about the 5 families which human G protein coupled receptors are split into. Okay, what we want to do in this video is firstly move on and talk about the Ballesteros Weinberg numbering system. And this really had to come after our discussion of the 5 main families of G protein coupled receptors, because it's dependent, the way this works is dependent on which family you're in. Okay, so this is the Ballesteros Weinberg numbering system, and as I said in the previous video, if you read any published paper, published review or published paper on G protein coupled receptors, you can't get away from the Ballesteros Weinberg numbering system for the residues of the 7 membrane spanning alpha helices of the G protein coupled receptor. Okay, right, so let me just draw a very basic picture then again for our G protein coupled receptor. So here's the cell membrane, and here's our very basic picture of the G protein coupled receptor with its 7 membrane spanning alpha helices. So basically, the Ballesteros Weinberg numbering system is a way of numbering the residues within the alpha helices of a G protein coupled receptor. Okay, so this is the idea. Firstly, let's say we have a residue. It's always easier to work with an example. So let's say we have some serine in helix number 4. Okay, so the way you would name it is you'd put SER, which is the 3-letter code for serine. Okay, then superscript 4, so you put which membrane spanning alpha helix it was in. Okay, and then you put a point, and then it's the number after this point that I need to explain. Okay, this is the difficult one to understand. Okay, you put a number that tells the person where this serine is in the membrane spanning alpha helix. But how do you do this? Well, basically, what has happened is some people have sat down and they've gone through all of the G protein coupled receptors in each of the 5 great families of G protein coupled receptors, or they've got a computer to do it. And what they've said is they've looked for the residue in each membrane spanning alpha helix that is most conserved in each of the 5 families. So let's say we start off with the great main family, the rhodopsin family of G protein coupled receptors. Okay, so you go into this rhodopsin family of G protein coupled receptors, you go through all 701 G protein coupled receptors, and for the 4th membrane spanning alpha helix, you look for which residue is most conserved in all of those 701 G protein coupled receptors. I mean, it would be lovely if there was one residue that was present in all of them at roughly the same point in the membrane spanning alpha helix. That would be perfect. It's probably not going to be that case, so what you look for is one that's maybe present in 99% of them. Okay, and what you say is we'll call that residue number 50. Okay, so you say we'll call that residue number 50, the one that's conserved in the most of the G protein coupled receptors in that family, in that helix. Okay, and then what you do is you name all the other residues in this membrane spanning alpha helice according to where they are relative to this most conserved residue. Okay, so if I draw the alpha helix for a moment, okay, so let's draw the alpha helix like so. Here is our membrane spanning alpha helix. Okay, let's say this residue here turns out to be the most conserved one in all of the rhodopsin family G protein coupled receptors. You pretty much find this residue there all the time. You then label this one number 50, and the ones that were closer to the N terminus you'd count down, and the ones that were further towards the C terminus you'd count up. Okay, so if we're talking about the 4th membrane spanning alpha helix here, the C terminus is upwards. Okay, so the C terminal end is this side, and the N terminal end is downwards here. Okay, so that means that if we went upwards, if we went to the next residue along after this 50th one, we'd call that 51, then we'd call the next one along 52, 53, 54, 55, 56, etc. Okay. If we went instead down, we'd go 49 would be the next amino acid along, then 48, then 47, etc. And you'd continue on downwards, basically. Okay, so because this residue is conserved in as many as possible of the G protein coupled receptors in the rhodopsin family, that means that we have a good way of naming the residues in most of the G protein coupled receptors in that family. Of course, for the ones where it's missing, then that's more of a problem, but it's the best way we can do it, basically. It will work for the 99%. Maybe it won't work for the final 1%. Well, it won't work for the final 1% because obviously we won't have the residue that should be there for the naming system to work. Okay, but it's the best way we can do it, basically. Okay, so I'll go over that once more. For each of the 5 families, so we've taken the rhodopsin family, but you'd also have to do it for the secretin family, the glutamate family, the adhesion family, and the frizzled taste2 receptor family. And the reason you have to do it differently for all of them is that you can't find one residue that is going to be conserved hugely amongst all of those different families. Okay, so you have to do it separately for each of the families. And for each of the 7 membrane-spanning alpha helices, you go through all of the members of the great family and look for a residue that is most conserved in those alpha helices in all of the 701 G protein coupled receptors. So what you then get is 7 residues, 1 for each membrane-spanning alpha helix, and they might be dotted around all over the place, basically, like so. And these are the ones for all G protein coupled receptors in that family, you are now going to label all the other residues of that membrane spanning alpha helix relative to this really conserved residue, basically. Okay, so the ones that are more towards the C-terminal side of that helix, they will be counting, you'll be counting upwards from 50 to get to those, okay? And the ones that are more towards the N-terminus, you'll be counting downwards to get to those, okay? So what you might see is something like serine 453, okay, 4.53 rather. And what this means is that you've got a serine in membrane-spanning alpha helix number 4, and it's 3 residues on from the most conserved residue in helix 4 for this rhodopsin family of G protein coupled receptors. Okay, right, so that is the Ballesteros Weinberg numbering system. Okay, what we want to turn our attention to now is discussing diversity in the structure of G protein coupled receptors. So we've already discussed how between these 5 great families of G protein coupled receptors there is huge diversity in the different amino terminal domains that they have. Okay, what I now want to discuss is firstly there is intra-family diversity. Okay, so within the rhodopsin family of G protein coupled receptors, for instance, there are G protein coupled receptors which have very different amino terminal domains. An example that I'm going to give is the sphingosine 1-phosphate type 1 receptor. Okay, then what I want to move on to is diversity in the transmembrane domain. Okay, and how the membrane spanning alpha helices can be different in their secondary structures. So I want to talk about proline kinks and also pi helices. And then we'll turn ourselves onto activation of G protein coupled receptors. Okay, so firstly then, let's talk about this intra-family diversity in the amino terminal domain. Okay, and the receptor that we're going to look at, which is a member of the rhodopsin family of G protein coupled receptors, is the S1P1 receptor. Okay, so what does this stand for? This stands for sphingosine 1-phosphate. That's the S1P. So this is sphingosine 1-phosphate. And then it's the sphingosine 1-phosphate type 1 receptor. That's what this one down here is for. So type 1 receptor. Okay, so firstly, what is sphingosine 1-phosphate? Because that's evidently the ligand for this receptor. Well, sphingosine 1-phosphate is a lipid molecule. It's actually a phospholipid molecule because it's a lipid structure with a phosphate group attached to it. Okay, so it's actually present within the phospholipid bilayer. Okay, so I'll show this here. So here is the phospholipid bilayer, and you'll have these little sphingosine 1-phosphate molecules in the lipid bilayer. Now they'll be different from normal phospholipids in that they actually only have one hydrophobic tail, which corresponds to the bulk of the sphingosine molecule. Okay, and then they have the phosphate group at the front of them. Okay, like so. So this is my cartoon for sphingosine 1-phosphate. So it's a phospholipid that you can find little bits of it occasionally in the phospholipid bilayer. Okay, right. Now, there is a receptor then for sphingosine 1-phosphate present in the phospholipid bilayer, and this is known as the sphingosine 1-phosphate type 1 receptor, and it's an example of a rhodopsin family G protein coupled receptor. So let's draw it here. Okay, so once again, here is this bundle of 7 membrane-spanning alpha helices sitting in the membrane like so. Okay, and we'll have the gap down the middle between the ring, or within the ring of the 7 membrane-spanning alpha helices here. So 1, 2, 3, 4, 5, 6, 7. Okay, right. Now the key thing that I want to show here is that this sphingosine 1-phosphate receptor 1, it is different from the other rhodopsin family receptors that we've looked at before in that it has a lid. It has something that covers this extracellular entrance to the ligand binding domain. Okay, now why is it okay for it to have a lid? Well, because the ligand doesn't need to enter from the extracellular fluid into the ligand binding pocket here. Instead, the ligand is already in the membrane, so it needs to enter the ligand binding pocket from the membrane, and in fact it does so by moving between the 1st and the 7th membrane spanning alpha helice over here. Okay, so let's firstly discuss then what actually makes the lid. So partly it's the amino terminal domain and partly it's the extracellular loop 2. So let me show this here. So here we'll have the extracellular loop 2 here between membrane spanning alpha helix 4 rather, and membrane spanning alpha helix 5 here. Okay, and then here we'll have the N-terminal domain here that will be binding with the extracellular loop 2 and will also be contributing to forming this lid that covers the extracellular entrance to this ligand binding pocket here. So let's just label these things on. So this is the extracellular loop 2 here between TM4 and TM5 here, and here is the amino terminal domain prior to TM1 over here. And then, of course, here's the C-terminal domain down here. Right, so those two portions together make a lid in the case of the sphingosine 1-phosphate type 1 receptor. Okay, and that covers up the extracellular opening to the ligand binding pocket. Instead, what happens is the sphingosine 1-phosphate molecules, which I'll draw another one here. Okay, so here's the sphingosine molecule in orange, and here's the phosphate group at the head of the sphingosine molecule to create sphingosine 1-phosphate, which we'll just abbreviate down to S1P here, can enter between the first membrane spanning alpha helix, which is this portion here in turquoise, and the 7th membrane spanning alpha helix, which is this portion in blue here. Okay, so it will enter through a gap between the 1st and 7th membrane spanning alpha helix, and then it will bind in that ligand binding pocket basically, and then activate the sphingosine 1-phosphate type 1 receptor. Okay, so that's an example of where there is diversity even within a family. Okay, so diversity in the amino terminal domain within a family. Okay, so sphingosine 1-phosphate has a lid. Another example of a receptor actually that has a lid is the rhodopsin family itself. So, well, the rhodopsin receptor itself. Okay, again, the rhodopsin receptor actually already has its ligand attached inside it. Okay, or what's going to become its ligand rather. Okay, so I'll just say a little bit about rhodopsin now. Okay, so basically rhodopsin all the time has a molecule bound to it, which is called 11-cis retinal. Okay, but this molecule doesn't usually activate the rhodopsin receptor. Instead, when a photon of light hits the 11-cis retinol, what will happen is 11-cis retinol will photoisomerase and it will turn into all-trans retinal. And all-trans retinal now can activate the rhodopsin receptor, can cause the change in conformation that then allows the rhodopsin receptor to interact with heterotrimeric G proteins, and we'll see the heterotrimeric G proteins transducing proteins which rhodopsin interacts with later. Okay, but again, the molecule is already attached to the rhodopsin, okay, and it's a derivative of vitamin A, and vitamin A is a fat-soluble vitamin, so again, it doesn't need an extracellular opening of the ligand binding pore because the ligand is going to come in from the membrane again. Okay, right, at least when you're loading the 11-cis retinol onto the opsin, which is the name for rhodopsin before it's got the 11-cis retinol attached in. Okay, right, that's the sphingosine 1-phosphate receptor and this lid that you can have. Okay, what I now want to talk about is diversity in the transmembrane domains. Okay, so the transmembrane domains of G protein coupled receptors are not all identical. Okay, they generally become more conserved towards the intracellular aspect. Okay, because after all G protein coupled receptors do need to activate G proteins. Okay, so they generally have more similarities towards the intracellular aspect where they're going to be interacting with G proteins, but they can have large scale secondary structure changes basically, which is what I want to talk about now. Okay, right, so let's start off by talking about proline kinks then. Okay, right, so proline kinks in membrane-spanning alpha helices. Okay, so this is when the membrane-spanning alpha helix has a proline present within it, and this causes the membrane-spanning alpha helix to kink. Okay, so let me show this. So here is the alpha helix shown here, and I'm trying to show it as regular as possible. I'm failing a little bit, and then suddenly you'll come to a proline, and we'll say the proline's here, and then suddenly it will kink, and it will start going off in a totally different direction, like so. Okay, so where should I show the proline? I'll show the proline here. So here's the proline, and it causes this kink, and that then causes the membrane-spanning alpha helix, or indeed any old alpha helix. It doesn't necessarily have to be a membrane-spanning alpha helix. We're interested in membrane-spanning alpha helices since we're talking about the transmembrane domain of G protein coupled receptors, but it could be any old alpha helix. Okay, so you have a proline present within the alpha helix, and that can then cause this kink, and that's known as a proline kink. Okay, so G protein coupled receptors frequently have proline kinks, and they'll have them at different places within the transmembrane alpha helices. Okay, so this is one of the ways that the transmembrane domains of G protein coupled receptors violently differ basically in their secondary structures, but if you look at different G protein coupled receptors. Okay, so let me just show you the structure of proline, and then you'll understand why this might cause a kink like this. Okay, so proline's a weird amino acid because the amino group of proline is actually involved in the R group. Okay, so let me show you this, and I'll show you it as a residue, so I'll show it as though it's bound within a polypeptide. And so here's the amino group, here's the alpha carbon with a hydrogen coming off it, and here's the carboxylic acid group. Okay, and then it will be bound to the amino group of the next amino acid along. Okay, right, and then the R group of a proline consists of 3 additional carbons, like so, making up a loop here, with hydrogens coming off the carbons. Okay, so each carbon will have 2 hydrogens. So you have these 3 methylene groups, like so. Okay, so this is the R group of proline, and again I'll stress that no other amino acid that's used in proteins, apart from modified forms of protein such as hydroxyprotein, okay, which is big in collagen, has this arrangement where the amino group is actually going to be involved in the R group, and this is what causes then that kink, okay, because the protein has a very different structure to normal amino acids, and therefore causes the alpha helix to veer off in this opposite direction, basically. Okay, so that's protein kinks, and that's one of the things that can lead to the secondary structures of the transmembrane alpha helices being very different between different G protein coupled receptors. Another thing which can cause kinks, which is separate from protein, is what's known as pi helices. So let me just explain what a pi helix is. Okay, so I want to contrast a pi helix to an alpha helix, and basically you can suddenly have pi helices in amongst alpha helices, and when they end they can cause kinks, basically. Okay, so firstly let me discuss what an alpha helix is with you a little bit more. Okay, so basically, previously all I've said about alpha helices is that they are spring-like structures. Well, the problem is I'm going to now have to repair that definition, okay, because pi helices are spring-like structures as well. So what is the specific feature of these spring-like helices that makes them alpha? What's the alpha for? Okay, and the alpha tells you the number of amino acids basically that you have on each turn of the helix. Okay, so basically what it means is that if you have an amino acid, and I'll put the amino acid here, okay, here's its amino group, and we'll draw it as a residue. Here's the amino group, here's the alpha carbon with the R group, here's a hydrogen, and here's the carboxylic acid group. Basically, the amino group of this amino acid here is going to be binding through a hydrogen bond to the carboxylic acid group of an amino acid that was 4 amino acids prior to it. Okay, so if I draw this out, here comes the helix all the way around, like so, and now we need the next amino acid along. Well, we're actually going backwards here. Okay, the polypeptide will go forward, so we need the amino acid backwards from this. Okay, and it's 4 amino acids backwards from this. Okay, the 3 in between have just been denoted by a line. I'll rectify that in a moment. Okay, so here is this other amino acid below. Okay, like so. And basically, of course, this amino group here is going to form a hydrogen bond with this oxygen here. The oxygen will have a partial negative charge because oxygen has a much greater electronegativity than carbon, so the electrons in this bond are pulled towards the oxygen, giving it a negative charge. It's also got a lone pair of electrons which forms a centre of negative charge, so it's a perfect hydrogen bond acceptor. Okay, if we look at the amino group here, the hydrogen is going to have a partial positive charge because it's bound to the nitrogen, which has a much higher electronegativity than it, so it pulls the electrons in this bond towards it, and that hydrogen then is the perfect hydrogen bond donor. Okay, so what can then happen is you can get a hydrogen bond forming between those two structures, the hydrogen bond donor and the hydrogen bond acceptor there. Okay, and basically in an alpha helix you are going to have 3 amino acids sitting in between these two. So in this space where I've drawn just a line, that would be 1 amino acid. This space will be another amino acid, and this space here will be another one. So 1, 2, 3. And that's the rules that govern alpha helices. Basically, the carboxylic acid terminus of this amino acid here will be binding with the amino terminus of an amino acid 4 in front of it. So we'll go 1, 2, 3, and the 4th one is the one it will be binding to, and that pattern will continue on for every amino acid. So this amino acid here will be binding, will have its carboxylic acid group binding to the amino group of the amino acid 4 in front of it. So 1, 2, 3, and whichever one came after this here, it will be binding to, and that continues all the way on. Okay, now pi helices, it's exactly the same picture, except that now there are more amino acids on each turn. Basically now we need 4 amino acids in the middle here. Okay, so change this and now include a 4th one. So I might just put a line there. Okay, so now the carboxylic acid group of this amino acid here is going to be interacting with the amino group of an amino acid 5 ahead of it. 1, 2, 3, 4, and then the 5th one. Okay, and that means basically that pi helices are going to be bigger than alpha helices. Their loops are going to contain more amino acids. So if we have our alpha helix here, it will be the smaller version basically. Okay, so what can you have then in the middle of these membrane-spanning alpha helices? Well, you can have portions that actually for a moment are pi helices. Okay, so let me show this. So in these membrane-spanning domains, what can happen is here you're going along nice and steadily as an alpha helix, and then suddenly you can have a portion that suddenly becomes this pi helix, and then when you return back to alpha helix, it causes a kink. You go off in an opposite direction here. Okay, so that is another structure that you can find within these membrane-spanning alpha helices, and again it's a way that the transmembrane domains of these G protein coupled receptors can vary violently in their secondary structure in this way. Okay, so if you look at different GPCRs, some of them will have pi helices in some of their transmembrane helices, some of them won't. Okay, and that's a way that the transmembrane domains can vary. And as I said at the start, generally it's more towards the extracellular side of the transmembrane helices that you find variation. Okay, especially, for instance, if I get my picture back again, remember the rhodopsin receptors actually bind their ligands in this pocket between the membrane-spanning alpha helices. So evidently you're going to need variation there because we've got 701 different GPCRs, and many of these are going to bind very different ligands. Okay, so clearly you're going to need different binding sites for those ligands, and therefore the extracellular sides of these membrane-spanning alpha helices are going to have to be different, basically. Okay, so that's grand ways in which the actual secondary structure can change. Obviously the primary structure can obviously change, and that will affect what the R groups sticking into the ligand binding site are going to be, which is a key factor also. But this is a grander scale thing here. This is a change in the secondary structure of those membrane-spanning alpha helices. Okay, right. Then finally, the intracellular domain of the G protein coupled receptor, which consists of the 3 intracellular loops and also the C-terminal. This is more constant, okay, particularly the intracellular loops. Very much so, intracellular loop 2 and 3 are conserved generally better than any other portion of the G protein coupled receptor structure. Okay, so the cytoplasmic portion of the intracellular loops along with these, the cytoplasmic portion of the transmembrane domains and the intracellular loops, these are very conserved, and it's because these portions are going to be important in binding the G protein coupled receptor, particularly intracellular loop 2 and intracellular loop 3. Okay, right. So, now we've discussed diversity in G protein coupled receptor structure. What I want to now talk about is activation of G protein coupled receptors. Okay, so firstly, we are going to study what is known as the classical model of G protein coupled receptors. Okay, and the classical model is frankly wrong. Okay, but it is still a very helpful model, and all science is just models. Okay, and therefore we're going to start off with the classical model, and later we'll talk about biased agonism when it becomes important. Okay, so we'll build up to talking about that. Okay, so what is the classical model for how G protein coupled receptors work? Well, the classical model is that G protein coupled receptors are on-off switches, basically. The idea is that they can only be in 2 conformations. They can be in the first conformation, which is just known as R, okay, just standing for receptor, in its normal off state. Okay, so this is where it's not in a conformation where it can interact with heterotrimeric G proteins. The other state that they can be in is the state where they are active and they can interact with heterotrimeric G proteins. Okay, now the classical model is that these are the only two states that the G protein coupled receptor can be in, and that it's in an equilibrium between the two. And this is why I'm doing the classical model at all, to get this important concept across. Okay, that basically it's not a case that you actually need the molecule to bind, at least not in for some G protein coupled receptors. It's not the case that you need the ligand molecule to bind in order for the receptor to change into the activated state. Basically, receptor molecules are all the time moving from being in the inactive state here, and I'm just going to label this up as the inactive state, to being in the active state. Okay, so if you have collection of loads of these receptors, so let's say here's the membrane and we look up loads of these different G protein coupled receptors here. Okay, and they're all the same type of G protein coupled receptor. Okay, so these three are all identical. If you look at them, some of them will be in this inactive state and some of them will be in the active state. So a certain fraction of them will be in the inactive state and another fraction of them will be in an active state, and there will be a dynamic equilibrium whereby at the equilibrium proportion here, so let's say maybe the equilibrium proportion is 0.1, well the fraction 0.1 of them will be in the active state, so 10% of them will be in the active state and 90% of them will be in the inactive state. Okay, if this is the equilibrium proportions, then that means that when you have this sort of proportion, 90% in the active and 90% in the inactive and 10% in the active, then the rate at which you're having inactive ones turned into active ones will equal the rate at which active ones are turning back into inactive ones. Okay, so it forms a dynamic equilibrium, and therefore, although you're continuously changing back and forth between being inactive and active, the proportions of receptors that are either inactive or active at any one time will remain the same, hence why it's an equilibrium. Okay, so that's the dynamic equilibrium. Okay, right, so that, the main point I want to talk about the classical model for is to get across this concept that there is a basal activity in many G protein coupled receptors, i.e. some G protein coupled receptors, they can go into the active state even before any ligand is bound, and at any one time a certain fraction of them will be in the active state and will be capable of binding G proteins. And in fact, some people often then put a further arrow here and then have the state R star G, which is the state where the activated receptor has the G protein bound. Okay, so at any one time, a certain fraction of these receptors will be in the active state and therefore will be capable of activating G protein coupled receptors long before any ligand molecule has bound. So many G protein coupled receptors have a basal activity. It's not true of all G protein coupled receptors. Some G protein coupled receptors, the basal activity is so low that you might as well not even acknowledge that it's there, i.e. really to all intents and purposes all the receptors are in the inactive form and not in the active form before a ligand molecule has bound. But for many G protein coupled receptors that's not the case, and you do have a basal activity where a certain fraction of them are in the active state all the time and the other fraction of them are in the inactive state. Again, that's long before any ligand molecule is bound. Okay, so the next concept that I want to talk about is the concept of what an agonist is going to do, what a classical antagonist is going to do, and what an inverse agonist is going to do. Okay, right. So an agonist then is any molecule which binds to the G protein coupled receptors and increases the probability that the G protein coupled receptor is going to be in the active state. So basically an agonist molecule shifts this equilibrium so that you have a greater fraction of the receptor molecules at any one time in the active state compared to the inactive state. So maybe it might shift it down to 0.8 in the inactive state and 0.2 in the active state. So 80% in the inactive and 20% in the active. That has shifted the equilibrium towards the active state. Okay, that's what an agonist does. Okay, so I think I'll color code this. This is what an agonist would do, shifting it towards the active state and therefore increasing the activity of the receptor from the basal activity, so resulting in more heterotrimeric G proteins and other signaling pathways being activated, although we're talking about the classical model, so we'll keep it simple and just think about heterotrimeric G proteins for now. Okay, then there is the concept of a neutral or classical antagonist, okay, which really is what people mean when they just say antagonist, okay? Neutral antagonist just stresses the point even more, okay? So, a neutral antagonist then, and actually I should stress that even though we think of many molecules, many drug molecules, we think of as neutral antagonists, the actual chances that they are neutral antagonists is actually very low, and I'll explain that in a moment. Okay, so a neutral antagonist is a very rare molecule. Okay, this is a molecule which binds to the receptor and does not change in the slightest the probability that the receptor will be in the inactive or the active state, i.e. it does not change the fraction of receptors that will be in the inactive versus the active state at all. All it does is blocks a binding site for some other molecule which might have been an agonist. Okay, so that's what a neutral antagonist is. A molecule which doesn't change the fraction of receptors that are in the inactive versus the active state at all. Okay, and although we call a lot of drugs antagonists to certain receptors, they usually are not quite neutral antagonists. Okay, usually they are actually this third type of drug, which is an inverse agonist. Okay, and what an inverse agonist does is it actually does the exact opposite of an agonist. It favors the inactive state. It shifts the equilibrium towards the inactive state, so it might increase the fraction that are in the inactive state and decrease the fraction that are in the active state. Maybe it might move the percentage in the inactive state up to 95% and the percentage that are in the active state down to 5%. So it shifts the equilibrium in favor of this inactive state for the receptor. Okay, and actually many of the drugs that we call antagonists are not actually this incredible thing of a neutral antagonist, but rather are actually inverse agonists slightly. Okay, they slightly favor the inactive state over the active state, and that can lead to problems, okay, because usually the basal signaling of G protein coupled receptors is very important, okay, and if you lose that because you've put in an inverse agonist, that can lead to problems when actually all you were hoping to do was have the effect of a neutral antagonist and just stop the signaling from being elevated by endogenous agonists. Okay, so it's not a trivial difference between a neutral antagonist and an inverse agonist. And watch it when people say antagonist, okay, because often when we call drugs antagonists, we think of them as neutral antagonists, but often it turns out that they are usually actually inverse agonists rather than this perfect thing of a neutral antagonist. Okay, even though it's conceptually easier to think of them as neutral antagonists, and that becomes important when thinking about the side effects of certain drugs, notably the H2 antagonists that are used to control stomach acid levels, okay, in the treatment of gastric ulcers and things like that. We think of those as neutral antagonists. They are H2 antagonists, drugs like ranitidine, for instance. Okay, so an H2 antagonist, we would call ranitidine an H2 antagonist, but in actual fact, many of them turn out to be inverse agonists, and they remove the basal activity of the H2 receptor, and this can lead to the side effects of this drug and why it becomes very difficult to then come off the drug, why you develop tolerance to the drug. Because if you suddenly come off the drug, well, I've missed a step out here. if you give this drug, it reduces the vasoactivity of the H2 receptor. So what the cell does is it increases its expression of the H2 receptor. Okay, so if you then suddenly stop taking the drug, you have far too much H2 receptors on the surface, okay, which are now going to have their normal vasoactivity, so you end up with far too much vasoactivity of these H2 receptors, and that initially can cause major problems. Of course, the stomach will readjust gradually, but initially it can cause problems. Okay, so the distinction between neutral antagonist and inverse agonist does have clinical relevance. Okay, right. So we'll call it there for this video, and in the next video what we will discuss is the actual mechanisms, conformational changes underlying the activation of G protein coupled receptors, okay, and then we'll move on to heterotrimeric G proteins and how they bind to the activated G protein coupled receptor.